What is epigenetics? Well, let's see.
It's the study of how DNA interacts with many smaller molecules within cells which can activate or deactivate genes. These are the molecules which largely determine what will be the outcome of a certain individual. Their presence and concentration within cells makes all the difference.
So now, how does that work?
The genes in the DNA are read and transcribed into RNA, which is then in turn translated into proteins by structures called ribosomes, and it is those proteins which determine the cell's characteristics and functions. Epigenetic changes can boost or interfere with the transcription of specific genes.
The most common way this interference happens is that there are some chemical groups which attach to the genome. When this happens, the genome is said to be marked.
The set of chemical groups that are attached to the genome of a cell is called the epigenome. Of these, like the methyl group, inhibit gene expression by causing the DNA to coil more tightly, therefore making it inaccessible.
The gene is still there, but is redundant. Boosting the transcription is essentially the opposite.
Some of these chemical marks will unwind the DNA, making it easier to transcribe. This increases the production of the associated protein. These changes can survive cell division, which means that they could affect an individual for their entire life. Epigenetic changes are a normal part of development. The cells in the embryo start with one master genome. As the cells divide, some are activated and others are inhibited. Through this epigenetic programming, some cells develop into heart cells and others into liver cells. So each of the approximately 200 cell types in your body has the same genome but its own distinct epigenome. The epigenomes can also be influenced by the environment. Factors such as diet, chemical exposure, and medication. Environmentally induced epigenetic changes are a part of the reason why genetically identical twins can grow up to have different lives. Even some social experiments can case epigenetic changes. In one experiment, when a mother didn't give enough attention to her kids, the genes in the babies that help them manage stress were turned off due to the chemical changes induced in them by this environmental change.
Meanwhile, the new genome editing techniques are making it much more easier to identify which epigenetic changes really influence us for our disease and life. Once we get to know how these epigenetic changes influence us, we might be able to influence it too.
For example, in future, we might be able to make ourselves genetically immune to the viruses of COVID-19, which has created not only a major economic crisis in the world, but also a major cause of health disruption, the extent of which is still not known and yet to be determined.